Microorganisms are essential in ecological, industrial, and clinical realms, necessitating precise methods to quantify their presence. The enumeration of microorganisms, term microenumeration, constitutes a fundamental aspect of microbiology. This experiment explores techniques used for microenumeration, focusing specifically on the viable plate count using pour plate and spread plate methods. The viable plate count method serves as a pivotal tool in microbiological analysis, involving quantification of viable microorganisms through cultivation on solid media, resulting in visible colonies. In the pour plate method, a known volume of the sample mixes with molten agar and is put into a sterile petri dish, ensuring even distribution of microorganisms within the solidifying agar. Conversely, the spread plate method evolves evenly, distributing a fixed sample volume across the surface of a solidified agar plate. Colonies form signify viable microorganisms, enabling quantitative assessment of microbial population in the original sample. Understanding the core principles of this method is crucial. In the pour plate method, microorganisms embed within the agar, giving rise to colonies on both the surface and within the medium. In contrast, the spread plate method confines colony formation to the surface. The choice between these techniques depends on specific investigate goals, each offering distinct advantages and limitations regarding accuracy and precision. This experiment aims to provide practical insights into microbial enumeration by employing the pour plate and spread plate methods. Participants will gain hands-on experience to better understand the microbial load in a sample and the significance of selecting suitable enumeration methods based on the nature of the microorganism under scrutiny.
Use a sterile pipette to transfer 1 ml of the sample to the 10 power of -2 dilution blank. Take 1 ml from the 10 of -2 dilution and transfer it to the 10 power of -3 dilution blank for further dilution. Repeat the previous procedure to create a 10 power of -4 dilution blank. Transfer 0.1 ml and 1.0 ml from the 10 of power -2 dilution to plates 10 of power -2 and 10 power of -3 respectively using a new sterile pipette. Then transfer 1 ml to plates 10 power of -4 using a new pipette from 10 power of -4 blank. Pour melted agar into each label petri plate containing the diluted sample. Swirl to mix without spilling over. Replace the lid and let the agar solidify. Incubate the inverted plates at 30 degrees Celsius. After the incubation, the colonies should be counted. 1 ml of water sample was pipetted into the 10 power of -1 dilution tube. 1 ml from 10 power of -1 was transferred to 10 power of -2 dilution tube. 1 ml from the 10 power of negative 2 dilution tube was transferred to 10 power of negative 3 dilution. By using a new sterile pipette, 0.1 ml from each tube's and original sample were pipetted onto agar plates. The glass spreader was sterilized with alcohol and was flamed. Then, each sample was spread using the glass spreader carefully. All of the samples were sealed. The inverted plates are incubated at 36 degrees Celsius for 24 to 48 hours. After the incubation, the colonies should be counted.
This is the table for pour plate method. However, they are not statistically reliable because they are below 30. This is the table for spread plate method. The first dilution has a statistically reliable data, while the others are statistically unreliable. The concentration of bacteria in the original sample is 13,800 colony-forming units per milliliter.
The variations observed in colony counts between the pour plate and spread plate methods can be attributed to the distribution of microorganisms within the agar. The pour plate method allows for colonies to form in both the depth and on the surface, providing a more comprehensive assessment. There are some disadvantages to this method, as the hot agar may kill heat-sensitive bacteria, and the depth of the agar may result in the reduced growth rate of obligate aerobes. In contrast, the spread plate method restricts colony formation on the surface, leading to potentially lower counts. Overall, the choice between these methods depends on the specific objectives of the analysis. For example, the pour plate technique is typically employed to count the number of microorganisms in a mixed sample, whereas the spread plate may be favored in the case of small sample volumes. Considerations such as sample characteristics, desired precision, and potential limitations should guide researchers in selecting the most suitable method. The experiment not only enhanced our understanding of enumeration techniques, but also underscored the importance of selecting an appropriate method based on the nature of the microorganisms and the desired level of precision in microbial quantification.
This is the answers for questions asked. Please pause to read.